In this video, we're going to continue with the phases of the cell cycle in detail. So over here, we're going to take a look at interphase. Interphase is the first phase of the cell cycle. It's the longest phase. It's 90% of the cell's life. It's actually divided up into three parts. There's a part called G1, S phase, and G2. And really, you could study those in a lot more detail than we are going to do in grade 10. You see this in a lot more detail in university especially. But different things happen here. But overall, in general, what's happening is growth and preparation for division. So G1 and G2 is really about growth. It's your number of organelles are doubling in G1 and G2, and there's other things happening. S phase, the S over here, is called synthesis phase. That's where the DNA is being doubled. So your DNA is doubling. What's important to understand is if you have a picture of interphase, when you look at that picture, you have a nucleus there, and then you see your DNA in the form of chromatin. It looks like spaghetti. It's not yet condensed, as we mentioned in the previous video. So DNA can be condensed either tightly or not tightly. When it's not wrapped around protein tightly, it looks like little strings or spaghetti, and we call that chromatin. And if it is wrapped tightly, then we call it chromosomes. And again, there's still a nuclear membrane there, and there's also still a nucleolus. That eventually disappears. But what's important is there's a nuclear membrane and there's chromatin. And this is our parent cell, our original cell. And during interphase, you're going through this entire process, 90% of the cell's life, in order to basically double in size, because everything is being doubled within you. And you're also making checkpoints. You're also checking to see that everything is being done properly. If you didn't double things properly, if you made mistakes, you got to go back and fix those before you go into division. So here you can see a labeled image of interphase. You can see the chromatin over here. You can see that this whole thing is the nucleus. This is the nucleolus. This is the nuclear membrane. And then over here we have centrioles as well. The centrioles are also in this process. And remember, those are the little organelles that kind of move around and go to opposite sides eventually, but not right now. This is a picture of interphase under the microscope. So you can see here, the way you can tell is that you have a nucleus still, you have a nice defined nuclear membrane, and then it looks nice and smooth in here. And the reason it's nice and smooth in here is because your chromatin is so thin, you don't really see all the little individual worm patterns yet. So it's nice and smooth there. The chromatin, the DNA is still in the form of chromatin, and so that's how you know you're in interphase when you're looking at a picture under the microscope. And again, here's another example of taking a look at the interphase under the microscope. We have that in an animal cell and a plant cell. So here's our animal cell. You can see the nucleus is still there. The nuclear membrane is still there, and it still looks pretty smooth in here. There's no thick veins that you can see yet. There'd be a lot of thick veins if you were in prophase right now, but right now it's interphase because it's smooth in the nucleus. Same thing here. You can see here's a plant cell. Here's a nucleus, and it's nice and smooth in there, and you can see the nucleus is still round and defined. So that means that the nuclear membrane is still there, right? When it's still round and defined, it means the nuclear membrane is still there. So you should be able to identify the cell cycle under the microscope as well, not just in cartoon images. Before we go on to the next phase, just a little bit of a thought experiment. So let's say you had a parent cell with 5 DNA molecules. How many molecules of DNA will it have after interphase? So right after it's on interphase, how many molecules of DNA? Just think about that. So as you're thinking about it, remember, during interphase, DNA is doubled. So you should have 10 molecules after interphase. How many molecules of DNA will the cell have after it's divided into 2? So how many will each cell have? Well, when you think about it, if you had 5 and you became 10, when you divide, you should now have 5 and 5 again. And that's basically it. So you can see here, you start off with 5, you double in interphase and become 10. Then when you divide, each daughter cell should have 5 DNA. So the reason that you're doubling in interphase is because you don't want to lose your DNA. If you didn't double and you kept 5 at the beginning, when you split, you'll then have 2.5 each. And that means you're missing half your DNA, which is not good for an organism. So now we get into mitosis, the actual part where we're starting to move the DNA around. We're in the first phase of mitosis called prophase. Okay, prophase. So the centrioles, which doubled in interphase, these organelles here, they're going to start to move to opposite sides of the cell. So you can see that here. These centrioles, so this is interphase right now, these centrioles are moving towards opposite sides. So here's prophase. So I'm comparing interphase to prophase. So the centrioles are moving to opposite sides. And From the centrioles, you're going to see these little strings start to come out. These strings are called spindle fibers, and they're important because later, what we're going to see is they're going to attach to the chromosome. So in prophase, these strings, when they're out, they're going to attach to the chromosomes here. Okay, and that's another important point to notice too. Is in interphase we had chromatin, but as we go into prophase, the chromatin condenses. You wrap the DNA around protein more so you can better manage it to this form here called chromosomes. Okay, so this is now a chromosome. This is your DNA even more fully condensed. So we go from chromatin to chromosome, and this is specifically called a replicated chromosome, and we'll go over that in more detail why it's called a replicated chromosome. But right now it is a chromosome. It's a replicated chromosome. And the middle part here, that's called the centromere of the chromosome, and that's where the spindle fibers are going to attach. So spindle fibers will attach right there, right there, right there, and right there on your chromosomes during prophase. So again, our main events that are happening during prophase is these centrioles are moving towards opposite sides of the cell. Opposite sides of the cell, we call that the poles. The nuclear membrane, the nucleus, has actually broken down. So you'll notice that there was a defined nucleus here. Now there's no more defined nucleus. You'll sometimes see pieces of it here. You can actually see pieces over here. It's broken down now. And if you think about it, that makes sense because the spindle fibers have to attach to the DNA. How can spindle fibers attach to DNA if the nuclear membrane is in the way? So that has broken down, and that allows for the spindle fibers to attach to the chromosomes. On that note, the chromatin has become chromosome form, so more condensed. And those are the main events happening during prophase. If you look under the microscope, it looks like this. So this is actually two pictures of prophase. I know this looks like interphase here because you have a defined nucleus, but if you look, there's a lot of little worms here, a lot of little veins. That means you now have chromosomes. Because chromosomes are very condensed and thick, you can see them more under the microscope. So this would be like a very early prophase before the nuclear membrane has broken down. And then here we have a later part of prophase where the nuclear membrane has broken down and the chromosomes are basically free. And try to guess what these parts are. The yellow that was over here. They were all together here, and now the yellow's here and here. So yeah, if you said these are centrioles, they are. They're the centrioles that move to opposite sides of the cell, the poles, and they have spindle fibers coming out of them. Now, it's hard to see spindle fibers in this image here, but they would connect to the centromere of the replicated chromosomes. This is taken by a fancy microscope where you can add color, so keep in mind you wouldn't necessarily see it this colorful under a microscope that we would look at it in class. But you can definitely have these fancy microscopes if you go study biology in university in more detail. So that was prophase, showing condensed chromosomes, spindle fibers attaching to them, normal nuclear membrane, centrioles move to opposite sides. So before we go into the next phase in more details, I do want to talk about the idea of the replicated or the duplicated chromosome. So when we see the chromosomes first form, they are in this replicated format. It's basically just very condensed DNA wrapped around protein. And you can actually see these under the microscope because they're nice and thick. If we go back here, we have thick chromosomes, so you can see them under the microscope. Whereas in the interphase, it was smooth, so you couldn't see those. So the replicated chromosome has a middle portion called the centromere. That's where the spindle fibers attach. And there's two basically sticks to them. There's this and this. And so the replicated chromosome is made up of what's called sister chromatids. The sister chromatids are just DNA molecules. So this is a DNA molecule, and so is this. And they're basically identical to each other. So think of it as like this. If you had chromosomes in interphase, which you don't, you have chromatin, they would be just one of these. But because DNA doubles, it becomes two of these now. And that's why we call it a replicated chromosome. You just don't see the replicated chromosome in interphase because it's chromatin at first. So these are sister chromatids. They're identical pieces of DNA. So one replicated chromosome is technically two pieces of DNA connected to each other. And the reason it's two pieces of DNA is because DNA doubled during interphase. So these are called duplicated chromosomes, a replicated chromosome. This is one replicated chromosome. So become familiar with that term. We're going to be using that more often as we go throughout the slides. So right here, back to prophase, we have 1, 2, 3, 4 replicated chromosomes over here. 4 replicated chromosomes. And then they're made up of 2 DNA each. So this is 2 DNAs here, 2 here, 2 here, 2 here. That means if you think about it, there's 8 total DNA in this cell. But before interphase, there must have been only 4, right? So there's 8 total DNA, because you can see 1, 2, 3, 4 replicated chromosomes, 2 DNA each. So there must have been only 4 before. So the original parent cell is 4 DNA in this picture. Okay, keep in mind we are 46 chromosomes in our cells, so we would have a different number than this. Again, here's some prophase that you can see under the microscope, a less fancy microscope, but you can see the chromosomes are starting to get thicker here. So you can see, over here you can see chromosomes are starting to get thicker here. You won't always be able to see spindle fibers like this, but you can see thickness of chromosomes over here. Okay. Over here too, you can see these are thicker chromosomes. You can sort of start to see the nuclear membrane break, but this would be an early prophase where you can see those thick lines, and it's not as smooth as it was before in interphase. So an animal cell and a plant cell. If I do give you a picture like this in an assessment or ask you about it during an interview, you should be able to explain that it's interphase and provide me with clues. And the main clue being that you can see the thick chromosomes here, and maybe you can indicate that nuclear membrane is breaking down, right? So it's about staying calm and trying to find the evidence to support why you think it might be prophase, for example. So now we go to the next phase. Interphase and prophase, there's a lot more details because you're getting oriented to all the different terms, but now we're just kind of continuing with the process. So this is prophase, and we're going into metaphase next, the next phase of mitosis. And so metaphase starts with an M, and it really is the middle phase. It's the phase where the chromosomes are lined up in the middle. So these spindle fibers that attach to our chromosomes basically move the chromosomes around and line them up in the middle of the cell. The middle of the cell is called the metaphase plate. Sometimes you'll hear the term equatorial plate as well, but it's the metaphase plate. You can see it in this picture quite clearly. All the worms are just kind of bunched together in the middle here, and you can actually see the spindle fibers attached here, spindle fibers attached there, but the worms are attached in the middle, or the chromosomes are. Chromosomes are attached right in the middle there, and you can see there's 1, 2, 3, 4 replicated chromosomes, each of them being made up of sister chromatids, which is the identical DNA essentially. And that's metaphase. It's pretty straightforward. Everything moved in the middle. So you can see metaphase quite clearly here. It's a pretty easy phase to spot. Here we have an animal cell, and you can see the darkly stained areas. Darkly stained areas are your chromosomes usually, right? They're very thick, so they stain very well. They absorb a lot of the color you put in to see under a microscope. And so right in the middle there, and you can see the centrioles here, there with spindle fibers connected. But right there you can see the chromosomes. This is the entire cell border, right? The membrane. And then here you have your chromosomes. And over here you have your cell wall in the plant cell. It's harder to see the spindle fibers and centrioles here, but you can really see those chromosomes right in the middle, that thick band in the middle there. Okay. Now notice not all cells have to be in the same phase in an organism. You can see next to it here, it looks like this part of the cell is probably in an interphase because you have a nucleus there. It's nice and smooth. I don't see any thick lines there. So we just zoomed in to one particular cell of this organism, and it happens to be in metaphase over here, but not here. So that was metaphase. So here we have metaphase where we left off. All the chromosomes were in the middle. And now we're going to go into anaphase. Starts with an A. Think of it as the apart phase. If you look at the picture here, you'll see the band that was in the middle is now starting to separate. If you look at the picture here, you can see that the chromosomes are starting to move to opposite sides. So what basically happened is the spindle fibers are shortening, and they split the sister chromatid. So this is a sister chromatid, and so is that. They pull them to opposite sides of the cell. So the replicated chromosome, this one replicated chromosome, is being split, and now these are still called chromosomes now. They were called sister chromatids when they were together, but now that they're separated, they become their own chromosome. But they're called non-replicated or non-duplicated chromosomes because they're no longer made up of 2 DNA each. They're just one. So this is one DNA, 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 one DNA. And you can see that they're going towards opposite sides of the cell, towards opposite poles. So again, now that you have replicated, non-replicated chromosomes, moving towards opposite sides of the cell equally, right? 4 going this way, 4 going that way. So that's an equal amount going both sides. The cell should look more like an oval now. It shouldn't really have this much of a curvature yet into it. You're going to see when we get to the next slide, there'll be a deeper curvature happening. It should, in most pictures you'll see it just as a perfect oval. There might be some imperfections in there, but you'll see more of a perfect oval in other pictures. But the point is we have chromosomes over here, chromosomes over here. And we're going towards opposite sides of the cell in terms of our chromosomes, and it's equal amounts going to both sides, and the chromosomes are no longer replicated. They are called non-replicated at this point. They're no longer called sisters anymore either because they've been separated. They're only sisters when they're together in their replicated chromosome form. And also notice the way they're shaped. They look like a V shape sort of, or over here a V shape or a U shape. That's because they're actually being dragged. And it's almost like the sisters are saying, no, don't pull us apart. And they're trying to reach for their opposite friend over here. But they're being dragged from the middle to the opposite side of the cell. And that's basically the majority of anaphase. You will notice the cell is elongated though. So we say it's an oval because it elongates. Here it's more of a sphere. For the pulling action to happen and the splitting eventually later on, the cell does elongate. So you'll see that as a good indication that you're probably in anaphase. Again, chromosomes are being separated here, going to opposite sides right now. And we still see chromosomes. That's important to understand. They're just not replicated at this point. That's anaphase. So let's go here under the microscope. You can see the animal cell and the plant cell. The animal cell over here, you can see chromosomes going to this side, chromosomes going to that side, darkly stained. And you can see they're actually still chromosomes. You can see that they have these like little finger-like projections, so they're still thick. Because as I mentioned in the overview, eventually the chromosomes become chromatin again, and that doesn't happen during anaphase. So you still see those thick little fingers here telling you that you have chromosomes still, so you know you're in anaphase, right? And you can see the cell is more elongated than it was before. And here it's harder to see the elongation because you have a cell wall, because it's a plant cell, so that's more rigid. But you can see your chromosomes are going this way and that way, and you can still see their chromosomes because of those thick fingers, those thick lines are there. So you're definitely in anaphase and not the next phase we're going to see called telophase. So now we get to telophase. So here's anaphase and here's telophase. Try to look at some differences before I go over it. So some differences you might have noticed is, first of all, you'll see the cell is starting to dip inwards. It's starting to pinch inwards. So here you didn't really have much of a pinching, but here you do. So it's starting to pinch inwards because we're going to start separating the cell into two. Now that's not really telophase. That's a separate part called cytokinesis, which is splitting the cell into two. But the point is that telophase, which is part of mitosis, happens at the same time as cytokinesis. So as the telophase stuff is happening, which I'll explain soon, cytokinesis is starting, and you can see the cell pinching more. You can see that pinching happening here. At this point in telophase, the chromosomes have reached opposite sides. In anaphase, they were traveling towards opposite side, but in telophase, they've reached the poles, they've reached their opposite sides, and 4 came here and 4 came here. So either, it doesn't matter what the number is, the point is that equal amounts came here and equal amounts came here. And you'll also notice that the nuclear membrane starts to reform. The nuclear membrane starts to reform around them. You don't really see that here, it's hard to see here, but you do see a nuclear membrane that will start to reform. And what you also don't see here is that eventually this chromosome is going to start becoming chromatin again. You can sort of see it over here. It's less defined, it looks more smooth now. So the chromosome is starting to become chromatin again because we're starting to go back to our original cells that we were before, or cell that we were before. So you can see this in these two pictures here. This is the animal cell. You can see the pinching start to happen in the middle. That's cytokinesis overlapping or happening at the same time as telophase. You can see the chromosomes are on opposite sides now, and you can see they're a bit more smooth. They're less finger-like. There's no more projections coming out. They're becoming less thick, so that means they're becoming chromosomes again, or chromatin again. And you can see that in the plant cell as well. You don't see a pinching because there's a cell wall that's there, but you can see sort of another little cell wall starting to form here. So in plant cells, they don't really have a pinching. they instead have another little cell wall start to form, which is kind of neat. And you can see the chromosomes reached opposite sides, and they're also more defined and smooth here, right? You can see that it's becoming chromatin again. So that's how you know you're in telophase in those situations. So again, if I provided you with a picture like this, could you tell me it's in telophase? And could you give me some clues? So could you say, oh, it looks like there's a new cell wall forming. It looks like the chromosomes are becoming chromatin again. Over here, if you had to give me some clues, it looks like chromosomes are becoming chromatin again, less thick. You can see there's a little pinching happening in the middle, and we'll see that term, what that pinching means soon. But can you provide me with evidence to support your answer as to why it's in a certain phase? And then finally, we get to cytokinesis. So I did mention that cytokinesis happens as telophase is happening. They overlap. But we're going to talk about cytokinesis by itself. So cytokinesis is actually where it's technically a separate part of cell division. So you have interphase, mitosis was the prophase, metaphase, anaphase, telophase is moving all the DNA around. And then cytokinesis is actually just splitting the cell up, but they kind of happen together. Cytokinesis overlaps with telophase. So you can see here that these two daughter cells were together, and now they pinch into two separate daughter cells. And the daughter cells are identical to each other and to the original parent. And you can see, at this point, once cytokinesis is done or almost underway, the DNA is definitely chromatin again, right? You're definitely back into chromatin. So here we can see kind of the early parts of cytokinesis happening with that pinching. And then we have two identical daughter cells. So just before we jump into any conclusions, I want to compare the animal cell mitosis and plant cell mitosis. So in this video, we saw the main phases of cell division. We went from interphase to mitosis to cytokinesis. In the next video, we're going to go and take a look at some main differences that happen between the animal cell and the plant cell. And a lot of those main differences really happen at the level of that cytokinesis area.